So today I'm revising the biology, so here are my notes. Let's study together and let's see how much I can finish in this video. So first of all, biology. Human body has 37 trillion cells. Bacterial eukaryotic cells are 10:1. Bacteria to eukaryotic, so bacteria are more than the eukaryotic cell. The cell theory was given by Robert Hooke, inventor of first crude microscope. According to that theory, all living things are composed of cells. Cell is the basic functional unit of life. Cells arise only from the pre-existing cells. Cells carry genetic information in the form of DNA. The genetic material passed on from parent to daughter cells could be prokaryotic, eukaryotic. Prokaryotic cells are the single cells, unicellular, do not contain any kind of nucleus, and eukaryotics are multicelled, then have enclosed nucleus in a membrane. The nucleus is surrounded by nuclear membrane or envelope. There are nuclear pores in the membrane which allows two-way exchange of the material. The genetic material regions are known as genes. If the cell has linear DNA which wounds around the histones, and these histones are further wounded around chromosomes. A subsection of the nucleus present which is known as nucleolus. These nucleolus also consist of ribosome RNA. They synthesize that. Then there is mitochondria. Mitochondria is known as the powerhouse of the cell. It has outer membrane and inner membrane. The inner membrane has multiple folds so that it has a lot of area to act, and these folds are known as cristae. The intermembrane space contains mitochondrial matrix. It does pumping of proton and release ATP synthase, which generate ATP during oxidative phosphorylation. The another role of mitochondria, despite providing energy and keeping the cell alive, is it is capable of killing the cells. So it has the property of apoptosis. Now comes the lysosomes. Lysosomes have enzymes that break down the waste product. Another thing which is important is endoplasmic reticulum. It has series of interconnected membrane that are actually contiguous with the nuclear envelope. Then the endoplasmic reticulum have smooth endoplasmic reticulum and rough endoplasmic reticulum. The smooth one lacks ribosomes and the rough one have ribosome. Ribosome permit the translation of protein for the secretion in the lumen, and the smooth one mostly used for the lipid synthesis. Then comes Golgi apparatus. These are basically packers and movers. They add up the carbohydrate, sulfate and phosphorus into the substance they are packing, and then package it and move it towards their destination. Then comes the peroxisomes. These are the cells containing hydrogen peroxide. This hydrogen peroxide is used for dissolving the fatty acids. Now comes the cytoskeleton. These are the microfilaments, microtubules and intermediate filaments. After that there is epithelial tissue. This covers the body and lines the cavities. It protects against the pathogen invasion as well. Then after that there is type of epithelium. First one is simple epithelium, which has one layer. Then stratified epithelium, pseudostratified epithelium, columnar epithelium squamous cells. Another thing which is important is prokaryotes. Prokaryotes contain bacteria, eukarya, archaea. Now what is archaea? Archaea earlier was the part of the bacteria, but then the scientists realized that archaea is the certain type of bacteria or similar to bacteria which can really survive in the extreme situations like salty, hot and cold environment. Bacteria have cell membrane and cytoplasm, and some has flagella and fimbriae. So archaea is basically type of a bacteria which can really survive in the really extreme environments. So it has been separated from the bacterial class and it has been given a different class known as archaea. Now the bacteria does essential function for human beings. They do produce vitamin K in the intestine, which is very important. So that's why for infants it is important to have vitamin K injections because they do not have any bacteria in them. Bacterial classification can be done on several types, but first of all there is rod, cocci, spirally. And then comes the aerobes and anaerobes. Aerobes can be obligate aerobes. Anaerobes can be obligate anaerobes. Facultative anaerobes, which are like can survive in the presence of oxygen. Then aerotolerant, they can also survive in the presence of oxygen, but they do not metabolize at all in the presence of oxygen or do not use the oxygen to metabolize. Then the obligate anaerobes are something which really, if the oxygen is present, they are killed. Then comes the Gram staining, which is used to identify crystal violet scales. So the cell wall, which is gram positive, consists of peptidoglycans and have the presence of lipoic acid. It is much more darker after the staining, so it is mostly the dark purple. The second one is the gram negative. After the staining, it stays pink. The pink stain is from the safranin. Then comes the genetic and growth of prokaryotes. There is a concept of binary fission. It is a simple form of asexual reproduction in which the prokaryotes replicate while they continuously grow in the size. The plasma membrane and cell wall begin to grow inward along with the midline of the cell and produce two identical daughter cells. E. coli replicates every 20 minutes under ideal conditions. Then comes the transformation. Transformation is the integration of the foreign genetic material into the host genome. Then there is conjugation, bacterial form of sexual reproduction, two cells form, conjugation bridge between them, facilitate transfer of the genetic material, and the genetic material is always transferred from male to female. Also, it is a unidirectional process. Then comes the transduction. Transduction is the genetic recombination process that requires a vector. A virus that carries genetic material from one bacterium to another. Then comes bacteriophage. These are the virus which infect the bacteria only, carry their DNA parts to the other bacteria during this. Then comes the bridge. The bacterial growth curve is an example of semi-log plot. The fact that the y-axis is logarithm means that a straight line as seen during the exponential process actually represents the exponential increase in the number of bacteria, not a linear increase. Semi-log, log-log plot as discussed. Then comes viruses. These are composed of genetic material. They can be linear, they can be circular. The linear have the DNA or RNA. Protein coat around the DNA. RNA is known as capsid. This capsid is made up of phospholipids. Envelope is very sensitive to heat and detergent. That's why it is important to wash off your hands so that any virus can be killed. Then also called obligate intercellular parasite, bacteriophages. Bacteriophages are the viruses which directly attack the bacteria. They are not harmful for the human being but for the bacteria only. So that was all for the chapter of cell. Now the next chapter will be the reproduction. I'll make another video for that. Thank you.